Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. In this particular module, we are discussing about the central dogma of molecular biology, and in this context, so far what we have discussed about the replications, and so we have divided the discussion about the replication, that it is going to be first we are going to discuss about the replication in prokaryotes, and then followed by that we are going to discuss about the replication in eukaryotes, because replication, the sole purpose of a replication is that it should synthesize the genomic content of that particular organisms in a limited time period, so that the organism should be able to divide and should be able to multiply its number, right. So, what we have discussed so far, we have discussed about the replication in the prokaryotic system, and what we have discussed that it is going to have the origin of replications, and origin of replication is going to be AT rich sequences, so that it is easy for the helicases and other enzymes what is been involved into the replications, and they should be able to melt the DNA and should be able to unwind the DNA very easily. And then following by that, we have also discussed extensively about the machinery what is required for the DNA replications in the prokaryotes. So, we have discussed about the DNA pol 1, pol 2 and pol 3. We have discussed about their structures, functions and all other kinds of features, and then apart from that we have also discussed about the helicases, DNAB, SSB structures, topoisomerases and other kinds of enzymes. So, in the previous lecture we have discussed all these aspects. Now, in the current lecture we are going to discuss about the special type of replications, and special type of replications what is being found into the prokaryotic system, and what are the differences and what are the different machinery what is required for performing these type of replications in the organisms. So, we have discussed about the replications, and we have discussed about the replication in prokaryotes. Now, we are going to see the special mode of replications what are being available or what are being found into the prokaryotic system, and how what kind of adaptation is happening into the bacteria, and how the different type of cellular machinery is involved. So, replication, if you see the replication has the very distinct 3 steps, right. You are going to have the initiation, followed by the elongation, and followed by the termination. So, if you recall, we have discussed that within the initiation, the cellular machinery will first identify the origin of replications, and there will be only one single origin of replication into the prokaryotic system, whereas there will be multiple origin of replication into the eukaryotic system. And once they have identified the origin of replication, then the initiation complexes are going to be formed, and the helicases and other kinds of enzymes will go and sit onto the initiation site, and that is how they are actually going to unwind the DNA. While they are unwinding the DNA, they will also going to ensure that the DNA what they are unwinding and preparing it for the replication is methylated. If the DNA is hemimethylated or unmethylated, then they will not actually going to initiate the replications. After that, it will enter into the elongation phase, where the DNA polymerase will go and sit and then start adding the nucleotides as per the Watson-Crick base pairing structures and so on. And then it will enter into the termination, and the termination is being done by the TER sequences, which are going to interact with the termination machinery, and that is how it is going to terminate. So, these are some of the basic structures or basic mechanism what is happening, but in some of the bacterial system you are actually going to have the replication in a special mode. So, one of the such special mode is called as the rolling circle replication or rolling circle model. So, in a rolling circle model, it is been found in the archaebacteria, bacteriophage, plasmids and the viral DNA such as the HHV, HPV, Gemini virus and the viral RNAs. In a rolling circle model, what you have is you have a double stranded circular DNA, and this is actually going to have the, so there will be a nick what is going to be formed onto the DNA, and then the machinery will actually going to utilize the information from the inner strands, and that is how there will be a synthesis of the DNA on one end, right, and it will be utilizing this information, keep rolling like this, and that is how they are actually going to start synthesizing. Once they will synthesize the one copy of genome, then it is going to be cut, and then this is actually going to serve as a template to synthesize the second strands. So, what are the different enzymes what are going to play a crucial role into the rolling circle model? So, you are going to have the rep A. So, rep A is going to initiate and nick the double stranded origins or DSO, right. Remember that this is the first event that you are going to have first form a nick structures, so that the machinery will enter into this structure, and it is going to utilize the nucleotide what is present onto the inner circle, and that is how it is they are going to synthesize this strand, and that is how it is actually going to make the multiple copies. Then you require the pCRA or plasmid copy reduced, and this is going to be a helicase which moves the nick strands. Then you require the DNA pol 3, and DNA pol 3 is going to have the 5 prime 3 prime polymerase activity, and it is going to be male replicase. Then you require the DNA pol 1, and the DNA pol 1, the function of the DNA pol 1 is going to be that it is going to remove the RNA primers, and then you also require the DNA ligase. DNA ligase is going to join the end to make the strand circular. Now, these are the some of the steps what you are going to perform. So, in the initiation, elongation and termination, right. So, in the initiation, the rep A is actually going to recognize the double stranded origin. So, in this case you are going to have the, for example, you are going to have the circle, and then in this, suppose this is the place, right. So, this is going to be a place where you are going to have the double stranded origin or DSO. So, this is actually going to go and allow the sitting of the rep A, and after recognition it is actually going to make a nick, right. So, it is going to make a nick into the upper strands, and so what is mean by the nick is it is actually going to make a break actually. Then in the elongation phase, the rep A stay attached with the nicked 5 prime phosphate end. So, it is actually going to make a cleavage, and it is going to hold the DNA, right. Whereas, the 3 prime end is free, acting as the DNA pol 3 primers, and the DNA pol 3 act in a 5 prime to 3 prime direction, produces the multiple concatemer, multiple single stranded copies of the original DNA series. To increase the efficiency of pol 3, one replicase pCRA is incorporated before it unwind to the double stranded DNA. And then in the termination state, you are going to have the rep A which is attached, cut the leading strand to stop the replication of that strand. Therefore, the second strand is to be left to synthesize. To replicate the second strand, the RNA polymerase or the primer synthesis, RNA polymerase and DNA pol 3 copies the single stranded origin and elongates. After replication, the DNA pol 1 remove the RNA primer and put the correct bases there, and then the DNA ligase will come and it will join the nicks to give you the double stranded circular DNA molecule. So, this is exactly what is going to happen, right. You have, this is the single stranded origin of replication, and this is going to be the double stranded origin of replication. So, when you have the, so in the initiation stage, your rep A is actually go and come and bind to this particular portion, and then it is actually going to make a nick. After making a nick, the rep A is actually going to bind the 5 prime end, whereas the 3 prime end is going to be free, right. And then on the free prime end, the pol 3 is actually going to bind, and then it is actually going to utilize this portion as a primer, right, this portion as a primer, and that is how it is actually going to start synthesizing the DNA, and that is how it is actually going to make the same copy, the single stranded same copy, right. So it is actually going to make the single stranded copy. So it is going to run like this, and that's how it is actually going to make the single stranded copy. So one strand is going to be synthesized fully, right. So for example, it goes like this, and one single copy is going to be synthesized, and then rep A is going to cut the strand, and DNA ligase is going to join the links. So it's going to have the double stranded, and it's going to have the other double stranded DNA molecules, and the single stranded DNA molecule is produced, which will be synthesized in a double stranded molecule using the similar kind of steps. So it's very clear that in the double stranded origin of replication, the rep A is going to bind, and then it is going to make the nick, and that nick is going to serve as a primer for the DNA pol 3, and then the DNA pol 3 is going to synthesize, and that's how it is actually going to make the single stranded concatemer, and these concatemer are going to be cut, right, once they will one round is over, right, and that is going to be double stranded simply by the DNA pol 1, and that's how it is actually going to have the double stranded circular DNA molecule, the daughter DNA molecule, and the other molecule is also going to be utilized. So in this one, you are going to have the two strands, right, this strand and this strand. This strand is going to serve the template to synthesize the outer strands, and afterwards, once the replication is over, then the outer strand is also going to be utilized to synthesize the inner strands, and that's how it is actually going to complete the cycle. One of the classical example of the rolling circle model is that it is going to be present in the some of the E. coli species, right. So you're going to have the donor cell, you're going to have the recipient cells, and once they will going to form, there will be a conjugation, and then after the conjugation they are going to exchange the genetic material. So how they are going to exchange genetic material is that the donor cell is actually going to have the rolling circuit model, right, and because of that its genome is going to be replicated, and it's going to have the concatemer, right. So this single stranded DNA is going to be transferred to the next recipient DNA, and that is going to be present to the recipient cells, and then the circular and the double stranded plasmid is produced by the new cells. So this single stranded DNA is actually a single stranded circular DNA is going to be then utilized as a template by the recipient cells, and it's going to make the double stranded DNA, and that's how you're actually going to have the plasmid, and that's how you are actually going to have the exchange of genetic material, and that's how you are going to exchange the phenotypic feature. For example, if this bacteria is ampicillin resistance, right, and this bacteria is ampicillin sensitive, then this bacteria is actually going to provide that resistance through the rolling circle model and provide the DNA responsible for that, and this DNA is going to provide the ampicillin resistance even into the donor molecule as well. So apart from the rolling circle model, you are going to have the another kind of replication mode that is called as D-loop formation or the D-loop replications. This replication is found in the small circular and organellar DNA. For example, it is present in the chloroplast and mitochondria. Remember that the chloroplast and mitochondria, although they are present within the eukaryotic cell, but they are not eukaryotic in origin, right. They are being prokaryotic in origin. So they follow many of these features what is present in the bacterial cell, right. For example, they have the circular DNA, they have their own DNA. So chloroplast and mitochondria are actually going to follow the mechanism what is being known for the or whatever we have discussed so far into the prokaryotic replication system, right. So where one triple stranded structure called a displacement loop is going to be formed, and mitochondrial DNA it is actually the 16.6 kb, which is consist of two strands, heavy strand and the light strand. So this is the heavy strand, the inner strand, and the outside is the light strands. It comprises a lengthier NCR or the non-coding region and act as the regulatory region. In this region, the mitochondrial DNA had its promoter for the transcription. One is the light strand promoter and the other is called as the heavy strand promoters or HSP. Conserved sequence MOXes, CSBs, and the termination associated sequences are also present. How? The replication terminate at the TAS is still not known. So, NCR also contains the origin of leading or the heavy strand. So, this heavy strand is called as the leading strand, whereas the light strand is also called as the lagging strand. So, origin of leading strand OH is on this heavy strand, whereas the origin of lagging strand is present onto the light strands. Now, what are the different key players which are involved into the D loop replications? So, you are going to have the DNA pol gamma. So, it is a main replicase or the polymerase. It has two subunit, polymerase A or pol A. It is from the DNA pol of the B A family. It shows the proofreading activity in the 3 to 5 exonuclease activity, and it is highly accurate. That is the one error in the 1 billion base pair. Then we have the pol B, which has the add-on subunit to improve the interaction between the pol A and the DNA templates. It increases both catalytic activity and the processivity. Then you also require the twinkle. So, twinkle is a hexamer helicase which needs the fork structure for being loaded and for start unbinding. Then you require the mitochondrial single stranded DNA binding protein. So, SSBs binds with the newly formed single stranded DNA to protect from the nucleases and maintain the single stranded structures. Then you also require the pol RMT. So, mitochondrial RNA polymerase synthesizes the RNA primer onto the displayed strand, as it cannot work on the single stranded DNA as a template. Then you also require the DNA ligase 3. So, DNA ligase joins the NICs in the new DNA strands. Then you also require the RNase H1 and MGM1. So, mitochondrial genome maintenance exonuclease 1 that helps into the primer removal after the replications. So, these are the steps of the D loop formations, D loop replications. So, you start at the OH site, the pol RMT starts making the primer onto the H strand to synthesize the whole H strand. So, you are going to have the genomic content in the form of the two circular strands, right. You are going to have H strand, which is on the blue one, and you are going to have L strand, which is the red one. So, in the first part, the on the OH side, which is present onto the H strand, is pol RMT is going to sit and it is going to start synthesizing the, it is going to start replicating that. Whole parental displaced H strand is covered by the single stranded DNA binding protein. The binding of the SSB stop the pol RMT for a random RNA synthesis onto the H strands. So, this is what is going to happen here. Then the twinkle, the helicase comes before the pol gamma to unbind the double stranded DNA to move the fork, while the SSB maintain the single stranded DNA, leading to the formation of the replisome. So, the replisome is going to be formed onto the DNA strand, right. After the 2/3 of the genome is synthesized, the replisome passes through the OL site or the origin of replication onto the light strand or L strand actually. There the parental single stranded H strand forms a stem loop kind of structures, right. So, this is a stem loop of structure, right, which is going to be formed. The stem loop structure stops SSBs from binding, thereby the one short stretch of the single stranded DNA in the loop becomes accessible for pol RMT to make the RNA primers. So, once you have this, it is actually going to stop the binding of the SSBs, and on the other hand, it is actually going to allow the pol RMT, or it is a RNA polymerase, to synthesize the primer for the L strand also. And then after 25 nucleotide of stem loop structure, the polymerase gamma replaces the poly RMT at the 3' end of the primer, which results into the synthesis of the L strands. H and L strands are replicated continuously until they reach to the termination sequences and forming a triple strand displacement loop D loop, and then the two DNA strands are going to be formed. So, this is what it is going to happen here, that you are going to have the L strand and H strand, and in the initial region the L strand on the L strands of the origin of replication, that is the OH side, pol RMT is going to bind, and that is how it is actually going to start the replications, whereas on the L side First, it is actually going to have the stem loop structure, and this stem loop structure is going to stop the binding of the SSBs, and it's also going to serve as a primer, and that's how it's actually going to allow the binding of the pole RMT, and it is going to synthesize the primer, and that's how it is actually going to initiate the synthesis of the L strands. And ultimately both the L strand dimer or the H strand is actually going to be formed, and that's how you are going to have the two double stranded daughter DNA. So, from this parent DNA you are going to have the two daughter DNA at this end. So, this is all about the DNA replications into the prokaryotic system. What we have discussed so far, we have discussed about the origin of replications into the prokaryotic system, and this origin of replication has the classical features of heterorich regions, and it is going to be recognized by the cellular machinery, and that's how the helicases and other cellular proteins are actually going to go and bind and unbind the DNA, and then it's going to be enter into the elongation phase, and in the elongation phase the DNA polymerase is going to synthesize the DNA strands, and it's going to join the nucleotide onto the incoming 3' site, and that's how it is actually going to synthesize, and once it reaches to the termination sites, it's going to be terminated by the TER sequences and so on. And in the current lecture, we have also discussed about the special mode of DNA replications. So, where we have discussed about the rolling circle model, which is very common in the bacterial system, and then we also discuss about the D loop formation, which is more common in the organellular DNA such as the mitochondrial DNA and the chloroplast DNA. So, with this small discussion about the prokaryotic replications, I would like to conclude the lecture here. In our subsequent lecture, we are going to discuss about the DNA replications in the eukaryotic system. Thank you.